Okay, hello everyone. I'm a fourth-year PhD candidate at the University of California, Irvine, and I'm here today to talk about the landscape genetics of pyrethroid resistance in Anopheles rabiensis across Kenya. And before I get started, I just want to point out that I'll be using the term resistance in two different contexts. The first is going to be in regards to insecticide resistance, and the second is going to be in relation to landscape resistance to gene flow. I'll do my best to clearly distinguish between the two uses as I go. So some background on the project is that Anopheles rabiensis have become increasingly abundant in Africa and are playing a key role in maintaining residual malaria transmission. While insecticide resistance in Anopheles rabiensis has remained relatively low with respect to Anopheles gambiae, there are recent reports suggesting the emergence of resistance in Anopheles rabiensis in Kenya. Which leads us to our central question driving the study, which is how does insecticide resistance spread in Anopheles rabiensis? To answer this question, we used the landscape genetics approach with the goal of quantifying the effect of landscape on gene flow. Landscape genetics first requires measuring genetic variation. So if we take a look at our hypothetical situation here, we have three populations that have been genotyped at various SNPs. You can notice that populations B and C differ by one SNP, whereas populations A and B differ by two SNPs. And next, we have to quantify landscape heterogeneity. So now our populations have been placed onto a landscape where a white cell indicates grassland, green forest, and blue water. In our case, we're mostly concerned with landscape heterogeneity between study sites since we're interested in how gene flows between populations. And lastly, we use statistical modeling to link the two to quantify the effect of landscape on gene flow. And given enough time, you could probably deduce from our oversimplified example that forest is a barrier to gene flow in this case. We test two competing hypotheses, the first being that biological or physical factors limiting continuity of mosquito distribution represent important gene flow barriers. Some examples of this are mountains, deserts, or land covers. Alternatively, we test that mosquito gene flow may be promoted by landscape characteristics that increase population connectivity. Some examples are roads and human distribution. And the significance behind the study is that if gene flow between populations is large, insecticide resistance could spread rapidly, and that knowledge of population genetic structure of Anopheles rabiensis is critical to understanding how insecticide resistance spreads. We collected Anopheles gambiae sensu latu larvae from 11 study sites across Kenya in 3 distinct geographical regions. Sites marked in green are in western Kenya, where malaria is hyperendemic. Sites marked in purple are in the Rift Valley, where malaria is epidemic or seasonal. And then sites marked in red are in coastal Kenya, where malaria is endemic. In the laboratory, we identified Anopheles gambiae sensu latu larvae to species using PCR. We genotyped Anopheles rabiensis mosquitoes using 10 microsatellite loci. Microsatellite loci were chosen from previous population genetic studies of Anopheles rabiensis in Kenya and surrounding countries, and were selected based on reliable amplification polymorphism, as well as an even distribution across the genome. A total of 542 individuals were genotyped, ranging from 30 to 58 individuals at each site. And lastly, Anopheles rabiensis samples were genotyped at the KDR allele. I created landscape resistance surfaces shown here using publicly available data. Landscape resistant surfaces are spatial layers that represent the extent to which a given cell constrains movement or gene flow. So for example, if we take a closer look at the elevation map in the top left corner, you can see that the lighter color indicates higher elevation, which in our case would indicate also higher landscape resistance to gene flow. And to give you a couple landmarks, we have the Great Rift Valley in the middle here, and then Mount Kenya over here, which is the highest point in Kenya. FSTs were used to measure genetic distance between populations, and we used two different methods for population clustering. We used a model-based approach of structure and an exploratory data analysis of a PCOA, or principal coordinate analysis. Landscape resistance surfaces were optimized using the resistance GA package in R. This method unbiasedly assigns landscape resistance values to categorical or continuous landscape variables. Next, landscape connectivity is measured using CircuitScape. CircuitScape relies on circuit theory, where a landscape is represented as a grid of regularly spaced nodes connected by resistors. Resistance surfaces determine the level of resistance between adjacent nodes. So if we take a look at my example here, a white cell would indicate no resistance. Pink is some resistance, and then dark red would indicate the highest level of landscape resistance. And the output is a pairwise measure that incorporates overall landscape resistance to gene flow between study sites. And this method is advantageous to the traditional least cost path because it considers all possible pathways between populations. And lastly, model fitness, or the fit of our genetic data, distance data to the landscape distance data, was measured using a linear mixed models effect with a maximum likelihood population effect. And this is preferred over the traditional mantle test because it accounts for non-independence among pairwise data. Okay, so here we have some results. These are the results of the structure analysis, where each individual bar represents one mosquito, and mosquitoes are grouped by the site in which they were collected, as well as the region they were collected in. And the different colors, yellow, orange, and blue, represent different likely ancestral populations. You can see that there's some population clustering between regions. For example, the orange cluster is much more prevalent in coastal Kenya than in western Kenya. But you can also see that there's no clear genetic structuring across Kenya, as seen by the blue and yellow cluster predominating in sites in both western and coastal Kenya. And you do see localized genetic structure. For example, Miwani has a much different genetic background than the rest of western Kenya. And lastly, I indicated study sites in which we detected the KDR mutation with a pink asterisk. And you can see that KDR was only detected in the 4 sites that have similar genetic backgrounds in western Kenya, suggesting that KDR is spread by gene flow. Next, we have a principal coordinate analysis, where you can see a similar clustering pattern to that observed in the structure analysis. For example, if we take a look at the populations in the blue cluster here, you can see our 4 similar western Kenya populations, but then you also see 1 coastal Kenya population clustered in there. And you also see coastal Kenya sites in the other 2 clusters as well. And this is likely due to a recent population expansion rather than lots of current gene flow. So now I'll present the results of the landscape genetic analysis. And since there wasn't clear clustering across regions and we only detected KDR in western Kenya, my analyses from here on focus on just western Kenya and the Rift Valley. So here we have a table of model fitness, or how well each landscape variable fit our genetic data, where delta AICC is a measure of model fitness, where a lower number indicates higher fitness, and omega is the likelihood that the model is the top model. So you can see that population density, distance to roads, and elevation all outperformed Euclidean distance alone. And since population density is our top performing model, I'm going to focus on that from here on. So here is a figure of the relationship assigned between human population density and estimated resistance. where you can see that a low human population density was assigned a high level of resistance, landscape resistance. And here's a map of the landscape resistance map based on our top model, and where a yellow color indicates more resistance to gene flow. And you can see that our Miwani site over here is surrounded by landscape barriers, as well as our Rift Valley sites, which is consistent with our structuring analysis. Okay, and these are the last results I'm going to present today. It's a map of genetic connectivity among our study sites based on our top landscape resistance model, where the yellow color indicates higher potential gene flow between study sites. And overlaying the map are the results of the KDR survey, where an X on the site indicates that node KDR was detected at the site. You can see that we detected both KDR 1014S and 1014F mutations in our study sites, and that KDR was mostly restricted to the westernmost sites in Kenya. And the last thing I want to point out is that the sites where KDR was detected are largely connected by the landscape. So in conclusion, there's no clear genetic structure in Anopheles rabiensis across Kenya, likely due to a recent mosquito population expansion. Local genetic structure within regions was detected, likely resulting from population founder effects. And finally, this supports the hypothesis that mosquito gene flow is promoted by near homogeneous human distribution, which is a spatial characteristic of landscape increasing population connectivity. And some implication these results have for malaria control is that KDR frequencies in Anopheles rabiensis are on the rise in Western Kenya, and that selection pressure coupled with gene flow will likely lead to increasing KDR mutation frequencies in Anopheles rabiensis, as was observed in Anopheles gambiae. And lastly, that KDR is most likely to spread between populations that are connected by a continuously high human population densities. And finally, I'd like to acknowledge our crew at the Kenya Medical Research Institute in Kisumu for all the work that they've contributed to this project. I'd like to thank my PhD advisor, Dr. Guiyan Yan, for his support and guidance along the way. I'd like to thank my lab mates at the University of California, Irvine for their support as well, and acknowledge our support from NIH. And I'll welcome any questions you have at this time.
We have time for a couple of questions.
Thank you. Nice talk.
Thank you.
My question is, have you done any simulations to check if genetic structure actually exists in Kenya, if you have this sample size to detect that genetic structure?
I don't believe so, no.
Just that is a nice thing to do in population genetics, and done in a lot of studies in population genomics as well, to check what is the required sample size to detect genetic structure
Right.
when it exists. It's just simulations. There's a lot of nice tools like CDPOP.
Right.
I would recommend.
Okay, thank you.
Yeah. Thank you for the talk.
Any other question?
Does the final heat map that you showed with kind of like the yellow distribution showing higher connectivity, was that informed by the population genetics also, or is that strictly landscape-based models?
The landscape model was informed by population genetics, so yes.
Okay, thank you.
Yep.